Hello guys, welcome back. I hope you guys had a great spring break. We're going to start off today with talking about HIV and AIDS. It's the first of two lectures about STDs, or STIs as I like to call them. I'll get into some of those details as well. But it's the first of the two lectures. I use HIV and AIDS as a whole lecture unto itself because it is so important in the global health arena. Again, from previous lectures, if you actually look at the numbers of people infected, it is relatively small compared to some of the other diseases that we discussed previously. However, when you actually consider the economic impacts that HIV and AIDS still has throughout the world, as well as the global funding arrangement for different programs, HIV/AIDS still has a huge component to the public health world in the terms of infectious diseases. So just a little bit of historical background. AIDS was first recognized as a new disease in 1981. It was first reported in the United States among homosexual males living in the Los Angeles areas that they found a certain type of pneumonia that you don't usually see in people, as well as a certain type of cancer called Kaposi sarcoma, which you don't see in normal healthy people. Usually very immunosuppressed people will have this, typically transplant recipients. But for the first time, they're starting to see this in what would have normally been considered healthy people, but they're primarily in the homosexual populations. A few weeks later, there was reports in San Francisco and New York about 26 males, also homosexual with the same conditions. They started to see this pattern, and they were figuring that there was something beyond just the homosexuality, that there was something underlying it as a disease that could have been an infectious disease. It was finally discovered by a couple of researchers, Luc Montagnier and Robert Gallo in the United States. 2 years later, in 1983, 84, winter of 83 into 84, it was finally isolated by those two scientists, and they realized that, yes, indeed, it was an infectious disease. And the infectious disease that they found that was causing this disease, this state called AIDS, or acquired immunodeficiency syndrome, was what they called HIV, or human immunodeficiency virus. The human immunodeficiency virus is a lentivirus. It's a member of the retrovirus family, which means that it has primarily RNA genome that replicates and turns into DNA. I'll talk about that in just a couple more slides. But HIV, they found, primarily infects CD4 positive T cells, macrophages, and dendritic cells. These are the immune cells themselves that they infect, so they actually have the ability, as they're replicating, to destroy the immune cells and thus creating immunodeficiencies. What characterizes HIV specifically is the high mutation rate. A mutation rate in the order of 1,000 times greater than bacterial DNA, and even greater in human DNA. And so you actually can get 10 to the 9 to 10 to the 10 virus particles per day production, and when you're dealing with a 3 times 10 to the 5th nucleotide mutation rate per replication cycle, you actually have a fairly significant amount of mutation. And that's what the major issue for developing drugs and vaccines against HIV is trying to figure out how to overcome this high mutation rate. They've done extensive studies on the genome of HIV, and they found that it contains 3 major gene products: the GAG, which is a nucleostructure protein, the POL, which includes the reverse transcriptase and integrase genes, and the envelope gene. Included in these other genes are a bunch of accessory genes which help in viral replication, budding, as well as certain activation markers for the ability to survive inside the body. The structure, three-dimensional structure of the actual protein structure, has also been elucidated fairly well, and you can actually see in the picture on the bottom right here that it contains two single-stranded RNA genomes. It has the matrix, reverse transcriptase attached to the genome, capsid, and then a bilipid membrane surrounding the whole thing with various capsid proteins, GP120 and 41 as the primary ones. I'm not going to go into huge detail about the life cycle. I did want to just emphasize that if you look at how the life cycle works here, the HIV genome primarily binds to a receptor called CD4, which is on those particular cell types I mentioned before, helper cells, macrophages, and dendritic cells. But it also has several other accessory proteins that it will bind to as it tries to enter the cell. Primarily CXCR4 and CCR5 are the two of them, and those two vary in varying degrees in the different types of cells between the macrophages, dendritic cells, and T cells. And the tropism, which cells it actually prefers, depends on what kind of mutation it has, and at various aspects of the infection cycle, it will change back and forth between going more towards CCR5 specific CD4 cells and CXCR4 specific CD cells. You don't need to know that in detail, just understand that the tropism that it goes for, it depends on those two particular receptors that it binds to. Once it enters into the cell, it will actually, the envelope from the virus will actually open up, allowing the the RNA to come in, and then the reverse transcriptase actually turns the RNA into DNA, and that DNA is now double-stranded, which will actually have a way to actually incorporate into the human genome and stay dormant. Once it actually incorporates into the human genome, what will happen is it'll utilize the human's machinery to produce new virus particles. And it does that by, if you remember from your basic biology class, transcription and translation. It'll be able to produce the proteins through translation, but the transcription itself produces new RNA, which is the actual RNA that the virus uses. It then uses the cell's membrane to bud out and create a new full virus particle, as number 7 demonstrates here. What even more complicates it, as I mentioned before, there are the high mutation rate leads into what they call clades. These are what I would call superfamilies or supergenotypes of the virus. So you have the individual virus, and then they have like certain types of mutation families. And you can classify these by letters, A, B, C, all the way down. And you can see that if you look closely, the serotype C, or clade C, is primarily in Africa, and serotype B is primarily in the Americas and Europe. Not exactly sure how this happened, but if you look at the pathogenicity of the individual serotypes, clades, it ends up being that the clade C, which is the African serotype, actually has the highest infectivity of all the different serotypes. Don't know why that is and why it's so African tropic. It could have something to do with the natural habitat of the virus and where it originated from, the phylogenetic history of it. But scientists are always still trying to figure that one out. So how's the disease spread? We mentioned before sexually transmitted. It's a sexually transmitted disease, but however, that's not the only way it can be transmitted. With monitoring and close evaluation, most of the other ways have actually been fairly limited nowadays. Exposure to infected blood and blood products can still be occurred. Contaminated clotting factors for hemophiliacs, transplantation, and those two are pretty much gone because blood is now pretty much throughout the world is screened for various transplantations as well as for hemophiliacs. So because of blood screening, you rarely ever see that type of transmission from HIV. However, what you still do see is a lot of contaminated needles among drug users. There's a lot of stigma that's related to drug use, and so because it's illegal in many countries, therefore it is something that the drug users try to hide and therefore tend to share needles, and that sharing needles can spread the disease. But also mother to fetus transmission. And this is an unfortunate one because they've shown that some of the first-line drugs, the AZT alone, can help reduce the transmission of HIV from the mother to the fetus fairly significantly. So the reason that this is happening a lot is because the mother doesn't know the status of her infection, and therefore screening pregnant women at the early stages for HIV is extremely critical and is not done enough in many of these countries. If you look at the life cycle, there's two main sections of the life cycle we're going to talk about. We're going to talk about the first section, which is the primary infection. This is when the person actually gets infected. And what you'll see is it's a typical infection profile for any particular disease. You get this massive increase in viral DNA, viral load, and then as your immune system starts to kick in, the viral load will come down, HIV antibodies will be developed, and also the viral proteins will come down. That happens in the course of weeks. So this is the typical primary immune reaction that you would have. What makes HIV different is that because it's not completely gone, because some of it actually was able to survive inside the T cells, macrophages, and dendritic cells, you still have a certain amount of viral load, and those viral load will slowly over time degrade your immune system, and eventually your antibody counts will come down, your viral load will come up, and you'll slowly develop HIV into AIDS as time goes on, and that happens in the course of typically in the years. You're starting to see a lot of people that can have disease profiles that can last 30, 40 years that they'll survive. With the new drugs that are available, HIV is now considered by many people to be a chronic disease, and it's being in healthcare systems. It's actually being routed into the chronic disease branches of their healthcare system rather than infectious diseases. Another thing to consider is individuals have different ability to react to HIV. There are certain types of people that are called long-term non-progressors, which means even without drugs, they can have the ability to not get infected, they are infected, but not develop AIDS. For some reason, they may have a certain amount of high load of T cells that are specific to HIV and B cells that are specific to HIV, so they can produce antibodies naturally that prevent the HIV from being able to spread faster. Having said that, even they will eventually be overcome by AIDS without any current treatments. So what is the definitions for the different terms? We talked about the T cell counts, and that's the primary way that you classify how bad HIV infection is. When you actually get infected, you start getting T cells that are reduced almost immediately, and it kills these T cells fairly quickly in some cases, but as mentioned before, some people, the long-term non-progressors, can go many, many years without having a problem. However, as time goes on, they'll eventually develop Category 1 HIV AIDS, which is less than 500 cells per mil of blood. And then as time goes on, it'll go down into Category 3, where it's less than 200 T cells per mil of blood, and that is the classification for AIDS. You're very, very sick by that point. And we'll get into some of that in some of the details as to what I mean by very sick. So the primary HIV syndrome, this is the first, that first couple weeks that I mentioned before. It's when you first get infected through the sexual contact, through needle sharing or so on. And again, this doesn't have much of a difference than a common cold or flu. You get some fever, rash, headache, diarrhea, sore throat, very non-specific type of symptoms that you would get here, and you probably don't even think of it as oh my God, this could be HIV. You would probably think you just got the cold, and for that reason you don't get tested. However, as time goes on, symptoms become more and more specific. Just talked about that. And when you start getting into the gradual declines for Category 1, where it's about 500 to 12,000 CD4 cell count, you start getting some of the stereotypical signs of HIV/AIDS. These are the early symptoms that are predictive that it's going to get worse. And again, these are conditions that you typically would not see in a normal person unless they've had some kind of other immunodeficiencies, such as a transplant where they're now on immunosuppressives. And you see things like oral candidaitis, thrush, oral hairy leukoplakia, or Kaposi sarcoma, some of the pictures down there. And these are some of the early signs that that you have a problem. And this is typically when most people will start to get tested, when they start to see this. Nowadays, the infectious disease doctors know this so well that they're not even, it would be the first test they would do. Back in the 80s, they were still questioning what was going on. As you start getting below 200, the person is considered to have full-blown AIDS. You start getting even more serious conditions such as toxoplasmosis, cryptococcal meningitis, CPC. If you get below 50, now these people are starting to get to the point where they are seriously injured. They're probably in the hospital in the last stages of dying. And you start getting conditions such as CMV infections, lymphoma, dementia, and typically death will occur when it hits below 50. Having said that, my wife is a physician, actually in Washington, D.C., and she sees many, many HIV/AIDS patients below 50. She said she's seen numbers as low as 7 that come in, and if you put them this is the first time they're coming in, mind you. This hasn't progressed over time. where they were taking drugs. They didn't even know they'd been sick until they got into the hospital with this massive problems. And so after you have them on 7, with a CD4 count of 7, you could put them on some drugs, and in many cases, actually, the people will actually come back and survive. And it's quite amazing to see that, how quickly the HIV medications can work fairly well. There are a spectrum of tests that can be done, and they depend on at what point of the infection cycle you're in. But for the most part, there's 3 major ones. You can do EIAs or ELISAs, enzyme-linked immunoassays. There's different types of rapid tests you can do, and there's Western blots. I'm not going to go into what these individual tests are. If you're interested, please feel free to look them up on Wikipedia or a generic immunology book. Early diagnosis in infants is usually looking at P24 surface protein, and the initiation and monitoring is usually done once you've actually been diagnosed. You're looking at CD4 counts and viral loads, and you want to make sure that the CD4 count stays high and the viral load stays low. Once you start seeing this shift between it, if one is, if CD4 is starting to go down and the viral load is starting to go up, if they're on medications, you want to start thinking about changing which medication they're on. There are a lot of different drugs available for HIV, and they come into several different types. And typically what is done is they'll merge several different types together to create what's called HART, or highly active retroviral therapy, and it'll usually include one RT, or reverse transcriptase inhibitor, and these interrupt basically the ability to make copies at an early stage, and it'll include also a protease inhibitor, the ability to prevent the production of the actual viral structure as it's trying to leave the cell. And it reduces HIV in the blood to undetectable levels. HART has been around since 1996 and has been basically the stalwart of the program since then. Here you can actually see where some of the drugs actually focus on. As I mentioned before, the retro-RT inhibitors prevent the reverse transcriptase to turn into DNA. The protease inhibitors prevent the proteins from being able to split to make the full capsid structures where the virus can actually leave the cell. There are many other types of brands of these drugs, and there's many different specific types of them. I mentioned the reverse transcriptase inhibitors, but there's also a fusion inhibitor which prevents the virus from actually entering into the cell. There's an integrase inhibitor which prevents the RNA, the newly formed DNA, after it's been reverse transcribed into being incorporated into the genome of the human. And there's different combinations of these. These are some of the drugs that are available. FDA-approved antiretrovirals. This is as of 2013, so there might be 1 or 2 new ones. I'm not exactly sure what the numbers are. If there has been any approved, I don't think there has been any approved in the last year or 2. So now let's get into some of the epidemiology of HIV and AIDS. It's still a major problem. As of today, there's almost 40 million people that have HIV/AIDS. And still that number hasn't changed since 2004. What's interesting is it's still the same problem. It's mostly in sub-Saharan Africa. The highest countries are Swaziland with almost 30%, and then even some of the areas in West Africa. Most of those countries where you see red here are in the 5 to 10% range. You'll get a couple that are much higher than that, in the 15 to 30, but for the most part, a lot of the countries have had some success in preventing HIV/AIDS, but you also have to understand these are people living with HIV/AIDS, and now are living, because they're on HART therapy, they're living longer, so the numbers aren't changing as a result. They might be on HART therapy, but they're still classified as HIV/AIDS positive, so that's why the numbers haven't changed. As they live out their natural course of lives, hopefully the normal 78 years, these numbers should go down since no new children are being born, and that's the hope that we have with the process. Okay, so when you're looking at the epidemic profile, you have to look at what kind of specific populations you have and the prevalence in those areas. So let's just take, for example, the extent of infection in a low-level area. If you look at a low-level infection, it usually has less than 5% prevalence within the key population. So that could be, the homosexual population, or the, it could be just the general population, in some cases women, and so on. And then you could also have, you want a less than 1% chance in the general population. And areas like this include areas like Middle East and North Africa. A concentrated infection would be you have greater than 5% with a less than 1% in the general population. And then areas like sub-Saharan Africa, you can have a greater or equal to 5%, and then even in the general population, greater than 10%. And that's a generalized high-level infection rate, and that's sub-Saharan Africa, for instance. Beyond just heart therapy, there are many, many different types of prevention programs and treatment programs. I just want to talk about this quickly. Each one of these I could give a whole lecture on, but let's just mention some of them. So we talked about the drug program, the drug treatment programs, but there's also treatment as prevention, and this is a new theory that's been going around, and this is kind of the big push in the global health community, in which you if you have a concordant partner. So if you are HIV positive and your partner is negative, you would actually treat both you and your partner, so that way that the partner does not get HIV. Having said that, you should still practice condom use and proper STI prevention programs. We talked about STI treatment before. Those are the heart therapy. Testing and counseling is extremely important. There's been a demonstration that showed that at each stage of the testing and counseling process, where you actually come in to get tested, you come back to get your results, then you come back to get counseling. At each stage of this program, the participation rate drops off. So a person might come in and get tested, but they never come back to get the results. If they come back to get the results, they may find out that they're positive, and they're so distraught that they may not come back for counseling because they're afraid to, or they're afraid that their husbands might find out, or things like that. So that's something that needs to be focused on. Condom use is still a major problem. It still has been increasing, but the education and condom use and education kind of go together. Education is extremely important. Sex education is still taboo in many countries. Blood screening has improved to the point where it's almost not really a problem. Drug and alcohol treatment, drug and alcohol abuse is strongly related, forget HIV, it's strongly related to so many other poor health outcomes. There's direct correlation between drug and alcohol use and increased chance of having HIV. And that goes along with harm reduction. Microbicides is a new thing that's being thought of. It's basically you add microbicides to the condom, or you can actually have a particular type of gel that you can apply. And it's still in the initial stages and some of the testing stages. There aren't too many that are approved. I don't think there's any approved at this point, but they are coming about. Maternal child testing is another one, and PrEP is also quite important. PrEP, by the way, is very similar to treatment as prevention. It's basically you take the pills as a way to prevent the treatment. You take the heart therapy as a way to prevent getting the infection. So successful national HIV prevention strategies, many of them have the same common threads. The biggest one that you have to be aware of is this high-level political leadership, civil society, and religious leaders. They all have to be working together. You have to reduce stigma, you have to have funding for it, you have to have awareness programs. Environmental and contextual factors, socio-cultural. At the community level, you have to be able to reduce the stigma of having HIV. You have to reduce as I mentioned before, the social factors such as drinking and alcohol. You have to have the economic and legal factors that can help people seek appropriate action if there's the ability to get the drugs, that you can afford the drugs, that there's a way that people who can't afford the drugs to still be able to get them. And then also especially pregnant women to be able to take the pills necessary to prevent the mother-to-child transmission. Open communication regarding sex to combat stigma and discrimination, mention that. Interventions based on epidemic profile, based on who the individual populations, the key populations are that are getting infected, to be able to target them. They are typically minority groups, social minority groups, marginalized populations. So special awareness at the government level needs to be taken, because at the community level they may be ignored. And so for that reason, these type of national programs are the ones that typically have the strongest programs. Talked about this. So we talked about some of at the different epidemic levels. What interventions can you do? And for an example, we're going to talk about condom promotion. If you have a low epidemic level, you kind of want to address market inefficiencies in condom procurement and focus on distribution to key populations. So again, at a low epidemic level, it's usually one or two small marginalized populations that have the disease, so you want to focus primarily on them. If you have a generalized high epidemic, you want to promote condom use and distribute condoms to free for everyone, because it's basically almost everyone in the community is at risk. So therefore, in that type of situation, you want to be able to spread it to everybody. And then also other things you have to consider is it's not just about prevention and treatment. People are still going to get eventually AIDS, and they're going to need care. And you have to have strategy for end-of-life care, which include home-based care programs. A lot of cases you have community health workers that'll come in and help out people that are infected, wash them, clean them, make sure that they're fed. And then also pain management, inexpensive options, because this is typically a neglected aspect of HIV care. The funding for these type of programs is scant. So inexpensive options are available, but significant barriers to access. When I talk about inexpensive options for pain management, a lot of times they'll give Panadol. If any of you know what Panadol is, it's basically Tylenol, but much, much weaker. And that's what they have for pain management. A lot of these people have bed sores because they can't get out of bed, and so it's pretty much not enough, but it's all they have. So being able to utilize the resources available is extremely problematic and critical in palliative care. Psychosocial support, people are dying, they're afraid, and so being able to give them that type of psychological support is also critical. And then nutritional support. I mentioned before, a lot of them are in bed, they're bed-stricken, their families might shun them, so they're not getting enough nutritional support, so you have to be able to account for that and take care of that as well. And then I mentioned, we talked about antiretroviral therapy. This is basically the key to being able to sustain programs, and being able to sustain a national program with antiretroviral therapy is extremely expensive, extremely complicated from a supply chain point of view, but it is basically all we have, and it's basically until there's a vaccine, if there's ever going to be a vaccine, it's all we really have. But basically, once people start antiretroviral therapy, they're on it for life. It's a complicated schedule, has a lot of side effects, and can be quite complicated. So being able to adhere to the therapy and making sure people adhere to the therapy is extremely important, because if they don't, eventually what will happen is you get resistance to the drugs, and you'll have to develop new drugs. And if all the drugs are gone, then we're going to have a major problem again. And able to do that, to watch out for resistance, you have to have a very strong laboratory monitoring to make sure that the mutations that are coming along are accounted for. Another thing you have to consider are co-infections. HIV is not in a vacuum of its own, and in fact, actually, HIV doesn't kill you. It's all the co-infections that'll kill you. The biggest ones being TB and malaria. Having said that, it's also the opportunistic infections that'll start to affect you, pneumonia, CMV, all these other types of things that'll eventually be the ones that you have to be, they'll eventually kill you. So being able to treat and prevent those co-infections before it becomes a problem for the person infected is extremely important. And then also a rigorous research agenda is necessary. As I mentioned before, there's no vaccine yet. Whether there's going to be a vaccine or not, I'm skeptical personally. I've been in actually the HIV vaccine world. I got my PhD in this. And the way I believe personally is that if they ever develop one, it's going to be similar to the flu vaccine, where they're going to have to constantly develop a new one because it mutates so quickly. Having said that, there are other issues, things that can be done in the meantime until there's a vaccine that's developed, and being able to evaluate programs for the most cost-effective and effective programs is probably up there. You know, distributing these drugs is probably close to $50 billion a year worldwide. And that's a huge amount of money from a global health commitment. And so being able to drive the price down while still providing an effective, efficient program is extremely important. And research to be able to do that is also necessary. The research to how to scale up the combination of different types of treatments, the combination of drugs, new drug development, and simplified regimens are all very key in this whole research agenda. So the last couple slides I want to talk about, if not the last slide, is just some of the HIV organizations out there, and there are a plethora of them. The biggest ones are WHO's, the United Nations AIDS Program, Global Fund for HIV/AIDS and TB and Malaria, Elizabeth Glaser Pediatric AIDS Foundation, PEPFAR, and the Clinton Health AIDS Initiative. These are just some of the biggest ones, but there's many, many others. RED are some of the funding, the funding organizations. USAID is big. There's many, many organizations out there for HIV/AIDS, and they all work together, not necessarily perfectly, but they try to work together to try to cover the world population that has AIDS, HIV and AIDS, and it's a mess. I mean, you're dealing with $50 billion to $100 billion a year, which is a huge amount of money, and a lot of organizations that have their own prerogatives. So trying to coordinate is, to be honest with you, a nightmare. And it is one of those things that, as I do it, I find it quite interesting to see how the relationships work. And I can only say, if you ever go into this field, while it is exciting, HIV/AIDS is a monster. So just be aware that, think about what you want to do and whether you want to be the big fish in the small pond with a smaller type of disease program, or if you want to be the small fish in the big pond, if I said that right. Small fish in the big pond like HIV, where basically you are just a voice in the chaos. Having said that, work is getting done. It's slow, but it is definitely getting done, and it's good to see that the results are there. And with that, I just want to thank you, and I hope you guys enjoy this week's questions, and we will continue next week.